All right, everyone, it's just me, so I guess most of you are going to watch this as a recording. Your test for Kelly Roberts' A&P Bio 207 Section 695 is going to be on Tuesday, the first hour of class, and it is over Chapter 10 and a large portion of Chapter 12. Because I was going to open this to questions, I just have a few things to cover for Chapter 12. If you do have questions, of course, you're welcome to email me or come by during an office hour of my schedule. But Chapter 12 is about the nervous system. And the very beginning of this system is, you know, just the big picture items, how it's divided, what are the branches. So the nervous system is divided into the central nervous system, the brain and spinal cord, versus the peripheral nervous system. Nerves tend to be one-way communication, so you have the afferents, which bring information to the central nervous system, and efferents, which take information to organs, glands, and effectors. So that's the efferents. Under afferents, it tends to be sensory. Efferents tend to be motor. So that's kind of the big picture, and then we start talking about neurons. So this first slide that I'm showing you here is to get into the microanatomy of neurons. This is showing you a multipotent neuron, multipolar. So that means that it has multiple dendrites. All right, multiple dendrites over here. And on the dendrites, that is going to be the primarily location for the neurolemma, where we're going to expect to find receptors for all the different neurotransmitters that tend to be how other cells talk to this neuron. The cell body, the central part here, we're going to see the nucleus, the nucleolus, neurofilaments, so cytoskeletal proteins, Golgi apparatus, mitochondria, and all the different organelles that, you know, are important for cell function. This nissl body is key because this is kind of a little factory of 3 ribosomes and endoplasmic reticulum, and then typically it's going to contain mRNA for important proteins that are usually going to make their way down towards the axon, and then that allows you to make vesicles for your neurotransmitters in the location of the axon and not necessarily in the location of the cell body. This is a key point here, the axon hillock, and we'll talk about this with electrical potentials. This is where the action potential threshold is critical, and therefore we initiate electrical signals down the axon or not. Okay. The neuron, like I've mentioned before, has a neurolemma. The axon lemma represents the cytoplasm in the axon area. And like your muscle, we're going to have around individual neurons, we're going to have endoneurium. Around a group of neurons, we're going to have perineurium, so they make kind of like a muscle fascicles. And then around a bunch of axons that constitute a nerve that's leaving or entering the spinal cord or the brain, you might see that it has epineurium. So that's the connective tissue around it. The synaptic terminals are similar to what we talked about in Chapter 10 with the neuromuscular junction. In this illustration here, the synaptic terminals, the synaptic knobs, the synaptic bulbs, it can be a few different names, are going to talk to another neuron. So this synapse is neuron to neuron. Okay. Now, the big part of this chapter is not every neuron actually looks alike. So your neurons are going to be either this anaxionic neuron, so it doesn't really have a true axon, but one of these areas will be the axon should it need to communicate with other cells. The bipolar neuron, you only really find these in the eye, okay? And what's unique about them is it technically kind of has 2 axonal projections. One is more of the dendritic piece that's bringing information from the multitude of dendritic hairs and then brings it to the cell body. And then the second projection is more of the true axon that brings the electrical signal to the synaptic terminals that then synapse with other dendrites and other cells and continues forward. All right, your unipolar are going to be lots of sensory. So in your afferent system, many of your sensory neurons take on this kind of motif. So the dendrites are technically might be modified receptors, such as your Pacinian corpuscles or your laminated corpuscles or your Meissner's or your Ruffinian cells, whatever that sensory type organ is. And the segment that carries the information towards the spinal cord is considered an axonal projection, and then it's another axonal projection. And typically in the sensory, this first projection here is the really long one. The cell body is actually located near the spinal cord, and all of these sensory neurons, their cell bodies kind of group together, and so it makes a ganglia or a gaggle. And they're called dorsal root ganglion because it's a combination of all those peripheral nerves that are bringing sensory information to the spinal cord, and all their cell bodies connect together. And then that second little axonal terminal is actually very short. So if you look for cells like this, look in your sensory branch, your afferent neurons carrying sensory information to the spinal cord from the periphery. And your multipolar neuron is the one we've seen in our tissue lab. We look for this one all over. This is definitely your motor neurons, okay? So your somatic motor neurons that reside with their cell bodies. This is in the spinal cord, okay? And this part is all in the spinal cord. The dendrites are getting information from another neuron in the pathway, okay? And then the axonal projects out of the spinal cord, travels into the muscle, all right? And then these synaptic terminals are going to synapse at a neuromuscular junction, okay? So we will find all of these different types of neurons all over, but we tend to see certain areas there are going to be certain neurons, so our motor tends to be more multipolar. Most of our sensory neurons tend to be unipolar. The bipolar are sensory neurons for the eye, and then these axonal are usually in the brain. The synapse. So the synapse is anytime we have a neuron talking to another cell. You can have neuron-neuron synapses, you can have neuromuscular junctions, you can have neurons and glands. So there's lots of different synapses, but in general, the synapse is going to have a presynaptic membrane, which is going to be the membrane that binds the vesicle and releases neurotransmitter. It's going to have a postsynaptic membrane, which is going to be where the receptors are located for given neurotransmitter, and the space is going to be the synaptic cleft. So these are the same terms we use to describe things in the neuromuscular junction. Now, the neuromuscular junction, the only neurotransmitter we talked about was acetylcholine, and motor neurons always used acetylcholine to talk to muscle fibers of the skeletal muscle organ system. But there are lots and lots and lots of different neurotransmitters. Some are considered like excitatory, some are considered inhibitory, lots of different neurotransmitters. Okay, just for example, we have acetylcholine, norepinephrine, dopamine, serotonin, to just be a few that I name off the top of my head. Okay, GABA, NMDA, lots of abbreviations because they're complex molecules. Okay, so this is what the synapse is. And the synapse is important because it's an electrical signal traveling here as an action potential. It's converted to a chemical signal with the neurotransmitter. And because neurons are going to be excitatory like muscle cells, it's going to maybe make another electrical signal on the neural lemma or the postsynaptic membrane. And that electrical signal, if let's say over time and frequency in multiple neurons, is enough to trigger a threshold at the axon hillock, it'll then trigger the neuron that's being spoken to, the postsynaptic neuron, to potentially continue to carry on that message as another action potential down its axon. Okay. Neuroglial cells. Excuse me. So the neuron is the functional unit of this tissue, and as we talked about in Chapter 4, the supporting cells are all thought together to be neuroglial cells. They're all called neuroglial cells. Now, the neuroglial cells, population-wise, we have 4 that tend to be found in the central nervous system, and we have 2 that are found in the periphery. Here are the specific names of those 4 neuroglial cells found in your central nervous system and what they kind of do. So the epidermal cells are highly branched processes, and they contact neuroglial directly. These tend to be our cells that are going to make, they kind of serve as epithelial cells, and they tend to be the ones that are going to make your cerebral spinal fluid, and they're going to line ventricles, and they're going to line chambers. Okay. Astrocytes are going to be large cell bodies with many processes. They get their name because they look like a star. And they are very important because they are going to help with the blood-brain barrier. So they're going to wrap around capillaries, and capillaries are continuous here, so they're not going to be very leaky, but these cells wrapped around those capillaries are going to make it even harder for things to leave the bloodstream and enter into the brain. The oligodendrocytes are going to be your myelination for your central nervous system. So all of your spinal cord, all of your brain, all of the axons that are myelinated, meaning they have fat wrapped around, are going to be because of oligodendrocytes. And they are different than how Schwann cells work for myelination in the periphery, and it's because of these cells and the differences of it and Schwann cells that we have issues with repair, injury repair, regrowth, and fixing of any problems that occur to neurons and damage that occurs to them. The microglial cells are actually going to be like your immune-ish cells. So because you have the blood-brain barrier, you don't really get macrophages and neutrophils, and you shouldn't get those immune cells really into your cerebrospinal fluid and into your brain. So these are kind of the cells that take that role then to play trash pickup and debris pickup and different functions of cleanup and protection. Okay. The satellite cells are going to also be known as amphicytes, and they are going to surround ganglia. So remember in the periphery I talked about you have your little like receptor. It brings its axonal process. It has its little cell body over here, and then it enters into the spinal cord. And all of the sensory neurons tend to have this gaggle outside the spinal cord. Right? And that gaggle is known as the dorsal root ganglion. And so the satellite cells are going to be in and around, and they are going to help protect those cell bodies of all those sensory neurons in that ganglion, so therefore they maintain sensory neurons and afferent ability. The Schwann cells are also called neuroglial cells, and they are going to give you a myelin sheath in the peripheral neurons. So they wrap around peripheral axons, insulate, and that insulation protects, supports, and helps with electrical signals being able to jump the long distances of the axons very quickly. Right? They are different from oligodendrocytes because one Schwann cell will only wrap around a very small portion of the axon, and it retains just the support and safety and health of that section of the axon. The oligodendrocyte is kind of like an octopus. It has its cell body in the space in between a bunch of axons, and then it has all these like arm-like tendrils that go and wrap around multiple areas of an axon or multiple axons. So the difference is if something happens to an axon, the oligodendrocyte isn't as great at helping repair and growth, whereas the myelin sheath of the Schwann cells, because each cell is there with its machinery and makeup to help make growth factors, you have better ability to regrow peripheral neurons that have been damaged than you do with central neurons. Okay, and this whole process of responding to injuries, the first thing that happens is Wallerian degeneration. So that means the axon distal to any of the injury dies, is removed, and then those Schwann cells, because they remain and because they have their cell bodies and they retain the pathway, they help stimulate the regrowth of the axon to go back into the tunnel that they are creating, and they help reconnect the axon to the target cell so it can reestablish the connection. And there's a picture of that in your book. Okay. Another big topic we need to talk about is the electrical properties of neurons. And I think Kelly has a lot on this on your test, understanding how the membrane and the electrical properties of the membrane work. All right. Now, the resting potential is also known as your transmembrane potential of a cell that doesn't have big movements of ions. Okay, so it's not conducting any electrical signals. Most of the time in the neuron, that can be anywhere from -70 millivolts, okay, and maybe even a little more negative, okay. For this transmembrane potential, they're in the neuron. It's primarily a function of sodium ions trying to go into a cell, okay, and it's potassium ions trapped inside the cell trying to get out, all right? You have some potassium that leaks out, and you have some sodium that might be able to leak in, right? And the transmembrane resting potential is predominantly influenced by the potassium levels, okay? Now, anytime the neuron is receiving information through chemical messengers, so neurotransmitters, and they're interacting with their receptors, those receptors can be tied to sodium channels, calcium channels, chloride channels, or potassium channels. And they can cause temporary, small changes from the resting membrane potential. Maybe they'll make it go -65, so they make it get a little bit less negative. Or maybe they make it more negative, so they make it go -75. All right? And those changes will kind of propagate and move from the point of origin, wherever that channel is, and move like a ripple of a rock being thrown into a pond, and will move down the membrane. And so the way you get an action potential generated is all of these graded potentials kind of summate or kind of build upon each other and cause at the axon hillock for threshold to be reached, which might be -55 millivolts, okay? Or they work together to make threshold harder to reach by making the resting membrane potential at the axon hillock move further away from -55 millivolts. So graded potentials are going to be the part of what's going on at the cell body, the dendritic end, and whether or not we get action potentials and we get the cells, the neurons, to continue to propagate a signal. And some things, you know, like pain, your endorphins and enkephalins, they kind of work to modulate and make it harder for pain signals to continue to be sent to the brain. So that way you're not in screaming, yelling pain constantly. It might dull for a little bit. Okay. Action potentials are going to be once we definitely get a definite change in resting membrane potential that is usually characterized by some form of rapid depolarization and then recovery to resting level. So there's a depolarization where the membrane typically goes from less negative to almost sometimes positive, and then it has to reestablish back down to resting transmembrane potential. Action potentials, once produced, they come from, again, the graded potentials, and at that axon hillock, you hit a threshold point, and then they propagate along the surface of the axon all the way down, and then can help at the axon terminal translate an electrical signal to a chemical signal and lead to the release of neurotransmitter. Okay? Synaptic activity is what happens with the chemical. So this is where that electric potential is transmitted to neurotransmitters, vesicle release. It travels across the synapse into the postsynaptic membrane, hopefully interacting with its receptors, and those receptors then open ligand-gated channels, maybe a ligand-gated sodium channel, allowing the membrane of that postsynaptic membrane to become more excited, more excitable. And information processing is the response or the integration of the stimuli at the postsynaptic cell. And so if we look at this from a picture, you typically have a resting potential, and that's kind of your starting point, okay? And as a neuron gets information from different cells and neurotransmitters are released, all right, those neurotransmitters are going to interact with their receptors at all these dendritic points and lead to, across all the different areas of the cell body, these little changes in resting membrane potential. Some might be little bits of depolarization. Some might be more hyperpolarization, so more negative membrane potential. And they are going to try to all work their way towards the axon hillock. And so as you get multiple signals or multiple places giving signals, some of them are going to build into more robust depolarizations, and some will build into more robust hyperpolarizations. And what happens here at the axon hillock, all these graded potentials come together, and either they cause an action potential or they don't. And then once you get an action potential, the axon's taking that electrical signal down to its end point, its synaptic bulb, and it's going to lead to the neurotransmitter release. So the electrical signal becomes chemical. And then what happens at this postsynaptic cell is going to be dependent upon what the neurotransmitter tells it to do. In a muscle, it's going to lead to contraction. In a gland, it might lead to release of sweat or oil or some secretion. You know, like salivary glands might, you know, release more saliva. So, and in other neurons, it might help the neuron become more excited and continue the message, or it might make the neuron less excitable and maybe dampen other messages coming from other areas of the body, like pain. Okay? So taking a deeper look, if this is our membrane, all right, so we would expect to find, you know, phospholipids here. As shown, there's some different proteins embedded in the membrane. The membrane, so here's the inside of our cell, here's the outside, and it's doing its job at being semi-permeable, so keeping certain things in, like big things like protein, and keeping certain things out, like some of these molecules out here, right? So in our cytoplasm, again, some of these big things like protein, nucleic acids, DNA, RNA, they're not going to leave the cell. And they're going to give the inside of the cell a lot of this negative products, a lot of negative molecules. And so what the cell does to compensate is tries to keep potassium ions, which are positive, to kind of keep all of this electrically balanced. But what ends up happening at the membrane is the inside of the membrane tends to take on an electrical property of being somewhat negative. The outside of the cell tends to have lots of sodium, and there's not so much sodium inside. And the sodium, and then the other thing that's sometimes out here is calcium, right, tends to give the outside of the cell a positive. So as this is showing you here, if I was to put some type of device that lets me measure the inside versus the outside, okay, I would see that because there tends to be more negative property towards the inside versus the outside, my resting across the membrane difference in voltage is a negative number. Now, anytime I move ions into or out of the cell, I'm going to now mess with this resting voltage, all right? And so what happens is the two things we pay attention to the most are typically if we let sodium into a cell, we tend to make the voltage less negative because we're adding more positives. If we let potassium out of the cell, we tend to make the voltage more negative because, again, we're letting a positive leave, and all these negative molecules that can't leave are getting a stronger effect. Anytime we do an action potential, right, we are going to have massive bringing in of sodium, and we're going to have the exit of potassium. And that's where that sodium pump comes into play that helps us reestablish, reset resting membrane potential by helping us force out sodium and force back in potassium. And because everything is forced, it requires energy. So looking at graded potentials, here is our resting membrane potential, and again, it's somewhere around negative 70 volts. Anytime you have a chemical stimuli that leads to something like sodium coming into the cell, or it blocks potassium from being able to leave the cell, anything that helps keep more positives inside the cell is going to let the membrane potential become less negative, and so it becomes a graded potential that is going to demonstrate in our little meter less negativity, so it tends to go upward. If the stimulus is removed and therefore we stop letting sodium in or we stop preventing potassium from leaving, we then reestablish and repolarize and come back to resting levels. If we apply a stimulus that, let's say, leaks out more potassium or lets chloride come inside the cell, we do something where we bring more negative in or we let more positive out, and we feel that the membrane gets more negative. And because it gets more negative, it's termed hyper, more polarized. And if that stimulus stops and we stop letting potassium leave or stop letting chloride out, we will recover. Okay? And if you look at graded potentials, this would be an EPSP, an excitatory, because it's letting us get closer to maybe threshold and bringing depolarization more likely to the membrane. And this would be an inhibitory signal because it's going to make us less likely to hit threshold. And the axon hillock gets all of these graded potentials, so EPSP and IPSPs, and based on them, will either hit threshold or won't. And so it will either be basically stimulated to continue the message or it won't. Okay, now what does an action potential look like? Okay, what you have is, again, you start off with resting, and through the neurotransmitter or through, in cases of the axon hillock, all of these graded potentials start to summate, start to kind of increase in strength because they're becoming more per second or more from different locations. You're going to then have the membrane reach this threshold, which is negative 60 millivolts. At negative 60 millivolts, we see a voltage-gated sodium channel open, and what that leads to is a massive influx into the cell of sodium channels. And so because all that sodium channel is coming into the cell, the membrane goes less negative, almost to and ends up being a positive voltage because there's more now positive inside, and it's switched over from being more negative on the inside of the membrane to more positive. At the peak, that voltage of positive 30 is going to close those voltage-gated channels for sodium. And it's going to trigger opening of voltage-gated potassium channels. And so now you have all this sodium inside and all this potassium so far and all these positives. The potassium goes out. And as the potassium leaves, you start to see positive charges leave, so the membrane is going to still work its way back towards resting levels. Okay? And actually, depending upon how long it takes, some of these potassium getting out might actually help cause the membrane to go a little bit beyond resting, and so it can hyperpolarize. Okay? So that's what those numbers represent. And then once we get back to near resting conditions, all of your potassium, all of your sodium channels are closed, and so you reestablish resting membrane potential, transmembrane potential, and it's ready to be hit with another action potential. Okay? One of the things to note is there is a time for every neuron where it absolutely cannot respond to any stimuli. And during that time, it's known as the absolute refractory period. And the main reason why it cannot respond is the sodium channels are open and they can't reopen, or they are truly closed and in a state of transition, all right, where they can't reopen again, all right? And so until this point is passed, you will not get a second action potential. The relative refractory period, some of these sodium channels have closed over here, okay? And so if you end up with a signal that is really, really strong, you could potentially open, you know, that small population of closed sodium channels and start getting another influx and another depolarization. But it's more challenging to get another action potential during the refractory period because you are starting with less available sodium channels, and your gradient, because all of your sodium hasn't been removed back outside of the cell, isn't as strong, that chemical gradient, okay? All right, so in summary, and again, this was meant to just be a quick review, what are some of the functional cells and components of the nervous system? So the functional cell, of course, is the neuron, and we have multiple types of neurons. They don't all look alike. The components of the nervous system, we have the neuroglial cells, and 4 types of neuroglial cells are found in the central nervous system versus 2 in the periphery. Components, so we have motor, the somatic motor system, we have the autonomic system, sensory system, all of the peripheral versus the central. So we have some different like flow charts of how the nervous system is arranged. We've gone through, again, the 4 in the CNS, the 2 in the PNS. Specifically pay attention to Schwann cells versus oligodendrocytes. That's a key difference because those cells functionally do the same thing. They insulate, protect, and help speed up electrical signals by allowing axons to jump electrical signals to the space in between the myelination, but they are doing it in a different manner, and that then lends to the periphery being able to recover from injury and the central nervous system being less resilient following injury. Review and describe resting membrane potential, graded potentials, and action potentials. So kind of briefly went through that. The resting potential is going to be the sodium-potassium setting up chemical gradients that there's high sodium out, low sodium in, high potassium in, low potassium out. You have a little bit of leaky of sodium coming in, a little leaky of potassium going out, and it tends to be negative 70 millivolts. Graded potentials are what occur, these IPSPs and EPSPs, because neurotransmitters are interacting with receptors and opening some changes in ion ability to come in or out of the cell. And then action potentials occur at the axon lock, and it's a summation, a coming together of all these graded potentials, and if they add together, summate to a point that they can trigger a threshold and therefore trigger the voltage-gated sodium channels to open, we get that nice, it kind of looks like a twitch from the myograph, but we get that nice reading on your electrical output of movement of sodium in causes the membrane potential to go from negative 70 all the way up to positive 30, and then we see the recovery back to resting from positive 30 back to maybe even negative 75, negative 80. There's a little bit of hyperpolarization. Some of your key neurotransmitters, we'll get into this a bit more as we go, but acetylcholine is huge in the neuromuscular junction with skeletal muscle. It's huge with parasympathetics. Norepinephrine is a big neurotransmitter for the sympathetic system. Dopamine, serotonin, serotonin, you know, a lot of people take antidepressants, so it's mood. And dopamine, if you watched Awakening, they talked about L-Dopa back in that time. So there's a lot of different neurotransmitters. Synapses can be between neurons and glands, and neurons and muscles. And remember, synapses are always one-way communication, right? And the generating of an action potential is the electrical becomes a chemical, and that's the neurotransmitter, and the neurotransmitter will then hopefully cause some type of graded potential, some kind of electrical event that leads to something on the second cell. All right, since nobody came, there's no questions, but good luck. I will see everyone Tuesday, again at 2 p.m. is going to be your test. And then at 3 p.m. to 3:50, you will have time to look over muscles and bones related to the axial skeleton. And again, I'm here for anyone that needs me, so don't be afraid to ask. Okay. Have a good weekend. See you on Tuesday.